Is Rakhi
Good
there?
evening, everyone. I welcome you all to the fourth session of online lecture series organized by Physiological Society of India Educational Committee, and we started on our 89th Foundation Day this online lecture series. Today we are going to have our fourth lecture. In this lecture, we are having topics to be discussed on excretory system with special emphasis to acid-base balance. This lecture series is planned to have both synchronous as well as asynchronous learning. Those students who are live, they can enjoy the lectures during the presentation itself live, as well as this same lecture will be present available at the YouTube, so that those who cannot attend this lecture series live, they can study, they can learn, they can enjoy these lectures later on in their free time. The Education Committee of the Physiological Society of India has taken this initiative mainly targeting towards the UG students. All the topics that we are discussing here is mostly to the targeted for the undergraduate students. However, PG students, they can also join and they can interact, because this lecture is live. You can put your queries, your comments on the YouTube comments. Dr. Sampa, you can mute yourself.
Yes, ma'am.
We can You can put your comments or queries, questions online.
No,
We will
ma'am.
be addressing those
After
if
Rakhi.
possible, whatever possible. In addition, you can communicate to the Education Committee of the PSI, Physiological Society of India, as well as the speakers. You can communicate and you can put your queries. Today, with the excretory system, the first lecture will be taken by Dr. Rakhide Sharma. She is assistant professor in Belda College, Department of Physiology. She completed her MSc PhD with physiology, and she is a experienced teacher. So I wish happy learning, and over to Dr. Rakhi.
Good evening and a warm festive greeting to one and all who are live today and who will see later on also. And thank you, Dr. Nayak, Professor Nayak, for this introduction. And I am grateful to all education committee members for selecting today's presentation, that is an overview on excretory system, for this first session. And the next session will be Dr. Bharti on acid-base regulation. So let me first present my slide today. Just share my slide. Can you see my slide, Dr. Nayak or Dr. Sarkar?
Yes, your slides are there on the screen.
Visible, okay? Okay, thank you,
Yeah,
thank you,
it's visible.
thank you. Thank you. And this is my college, just the photograph of my college. Let my student first understand that what do we mean by excretion, because that will give them much interest to go further on the slides. Like excretion, we know for all living being, it's a process to remove waste or excess water or electrolytes from our body. In plants also, we know that the plant excrete by the way of removing the leaves from and covers also. The following organs actually, mostly if we will see the human beings, it's these organs are involved in this process, mainly skin, liver, large intestine, lungs, and kidney. Skin is the largest organ of our body, we know, and by removing water, some of the electrolytes, and some of the metabolites, skin play role in excretion. Liver is the vital organ of our body, and it removes, we know that many toxic metabolites, toxic elements from our body, and especially it removes ammonia in the form of urea. And because we know that hepatocytes are the only organ where urea is synthesized, and that urea then comes into blood, and then it removed. And large intestine, it is the part of digestive system, and whatever we ingest, the unwanted part, especially the fibers and those which is not required, is removed from our body through anus. And so large intestine play a role. And lungs, we know that from for different physiological activity, we need ATP. And ATP is synthesized by metabolism, especially the glucose. And the end product of glucose metabolism is carbon dioxide and water. And this carbon dioxide is removed through lungs, and lungs play a very important role in that. And kidney is another important organ of our body, which plays a very important role in maintaining the homeostasis, that is the internal environment of the human being. And in today's next 40-50 minutes talk, we will mostly concentrate on kidney, that is renal physiology, because otherwise it's not possible to complete. And the if a student also, if they understand what is the purpose of excretion, is that we know that different nitrogenous product is metabolized in our body, especially just to remind you that fat and carbohydrate in some or other way can be deposited in body, but proteins, they are either utilized or they has to be removed from the body. And these proteins, as they have mostly amine groups and carboxylic group, they usually convert into ammonia very easily. And ammonia is a very toxic product. So our body, especially human being, will try to remove this. And how they will do? They, especially in our body, liver converts this ammonia into urea. And actually, in living beings, we have three types of nitrogenous waste product: ammonia, uric acid, and urea. Okay. In next slide, you will see that these are the invertebrates, aquatic species, which excrete ammonia from their body. And mostly mammals, mainly adults and amphibians and some marine species, we excrete urea. And the birds and snails and insects, they excrete uric acid. What is the cause? Why they do like that? Why there are three different products, nitrogenous products is being removed from the body? This is as per the requirement of the body. Like, especially if you will consider the birds or insects, they have very less water. They need to use very less water, because they have to be light. We know their bones are very light. They eat, they don't can keep foods very much. They don't have water very much. So they preferred uric acid, because uric acid can be removed in semi solid form. Okay. And in case of these invertebrates aquatic species, because in aquatic species we know that surrounding water is also saline, so to maintain their physiological function, their water of their body, the electrolytes of their body, their diffusion, they remove ammonia from their body. And for us mammals and these amphibians, we prefer urea, not the uric acid or ammonia, as they are toxic for us. And here is just one from one paper, same thing which I have shown in previous slide, that mostly cellular proteins or ingested protein gets hydrolyzed into amino acid, and these are used for growth and maintenance. And if they extra will catabolize and they will form bicarbonate and ammonia will get excreted for marine animals, and for some it get retained, and others like urea and uric acid for birds and terrestrial animals, they are need either excreted or retained. They can be retained. Just to give you an overview on the evolution of the organ which is actually serve the purpose of removal of urea, uric acid or ammonia, is this is the primitive excretory organ where the flame cell play an important role in the process of excretion or in the process of removal of the excretory product. In earthworm, this metanephridia and this nephrostome and then coelomic fluids, formation of urine and then removal. And in ants, the fluid filled coelom actually play a role, and these are the primitive tubules, Malpighium tubules, just to show you that primitive Malpighium tubules in the ants will look like this. And then further we will come to our presentation actually in the human being. So what is the learning outcome from the today's presentation is that the student will able to understand or know the anatomy and function of kidney. They will able to draw and label the structure of nephron if they will go properly with the slides today and will understand the presentation today. They will learn to draw the structure and understand the importance of juxtaglomerular apparatus. And they will also learn the role of glomerulus and the renal tubules in the formation of urine, and also to understand the process of micturition. Let's first see the what is the whole this structure of renal physiology is that we have a pair of kidney, we have a pair of ureters and urinary bladder and urethra. Okay, by which the urine, this is where the urine gets collected. From here the urine comes from kidney, it's collected in urinary bladder, and through urethra it comes out of the body. Just an simple overview of the evolution of the kidney in human system. In the fetus, they will just development is very slow with at different trimester with different weeks of development. We know that in fetus they start working, the kidney started working, they remove the fluid that comes into the amniotic fluid, but not the proper function is only happens once a baby get birth. And this is also that an overview that how the development of nephron, that is one of the structural and functional unit of kidney, that whole function of kidney depends on this nephron, is happening in system. Transverse section of kidney, it's a bean shaped structure, and it has a cortex part, and this is cortex part, and this is medullary part, and this is renal vein and renal artery. This blood supply we will see in the details, and these are called the renal pyramids. Okay, and this is renal pelvis. Here actually after formation of urine it comes into renal pelvis, and then through after the pressure increased here, the peristaltic movement start from here, and then urine comes into the urine through urethra to urinary bladder. Okay, and now this is the structure of the structural and functional unit of kidney, that is nephron. In individual nephron, we have almost 90 lakhs to 1.2 lakhs of nephron. So you can understand that this small kidney, pair of kidney, will have so many numbers of nephron. That is the purpose actually of kidney or this nephron is the purification of blood. So the whole blood needs to be purified regularly in our body to keep our internal media in normal situation, okay, in normal condition. So if you'll just see the parts, I will not go in this slide that blood circulation. You just see the structure, tubular structure, and the structure of nephron. This is called this structure, this endless cup-shaped structure is the Bowman's capsule, okay, and followed by these proximal convoluted tubules next to it, and then comes the descending loop of Henle. There is thin descending loop of Henle and thin ascending loop of Henle, then thick ascending loop of Henle, followed by distal convoluted tubules, and then collecting duct. I talked about so many parts, that is Bowman's capsule, proximal convoluted tubule, and this loop of Henle, descending distal convoluted tubule, and collecting ducts. All of these structure have epithelial cell on their wall, and since they do different function and they have different function with respect to reabsorption, so their epithelial cells also have accordingly have their structure. Epithelial cell means we know that the cell, it will have a nucleus, it will have different organelle, but the organization of organelle, if you will see properly, it differs in different area. Like this proximal convoluted tubule. This is the woman's capsule part. Okay, so main filtrate come here, and then maximum will get reabsorbed here. That process I will come. So as per function, since maximum reabsorption is happening there, these proximal convoluted tubules, the epithelial cell of proximal convoluted tubule have so many microvilli. I hope the student remembers that small intestine have large villi, large number of villi in individual epithelial cell. The purpose is to absorb nutrients as much as possible, because to increase the surface area of this. So similar the case here, since maximum reabsorption is happening in proximal convoluted tubule, so to increase the surface area, so maximum epithelial cell have villi here. And the other thing is, you just see that so much mitochondria. I will just like to draw your attention for individual cells like proximal tubule, descending loop of Henle, thick ascending loop of Henle, distal convoluted tubule, cortical collecting duct. So you just see the difference in the mitochondria. These differences are because of their function. The more the cells is participating in active transport, they are having more mitochondria. That we know it, that mitochondria is required for formation of ATP through oxidative phosphorylation, and that also required the oxygen. Okay, so they do this function active transport. That means active transport is very much here in proximal convoluted tubule, is very much here in thick ascending loop of Henle, is very much there in distal convoluted tubule. However, this active transport is less in descending thin loop of Henle, and also the cortical collecting duct. Okay. So here as the same, that previous viewer I will just showed you that these things will be more clear to you. This is the same. And now I'm just concentrating on this Bowman's capsule. And now I will tell you that this is Bowman's capsule, and the blood supply which is coming here through afferent arterioles will make almost 50 small capillaries here. This is called glomerular tuft, and they will collect into efferent arterioles and then come out. Okay, so this Bowman's capsule has epithelial cell. These capillaries have endothelial cell. So, and the things get filtered. The blood which is coming here is get filtered here. So who plays the role? What is the, what is their, uh, how this is happening? Okay, so just to, in the next electron microscope structure, just one figure to show that this is one capillary, these are endothelial cell, and these are fenestrated region, and these are the epithelial cell or the modified epithelial cell, which we call here as podocyte food processes, and these are the slits. This fenestrated is larger actually. It's almost 90 to 100 nanometer, and they are almost four, maximum 4 nanometer. So whatever things get filtered from here will get come into the glomerular filtrate, which are less than size of 4 nanometer. Okay, so when blood pass through this, the size, the molecules which are in the blood less than 4 nanometer size will get filtered here, and more than 4 nanometer size, like high molecular weight proteins, albumin and globulin, and blood cells, they do, in normal condition they don't get filtered out here. Okay, so another thing to mention is that the nephron in uh, in our system is two types of nephron. One is cortical nephron and another is juxtamedullary nephron. We have 85% of cortical nephron, okay, and 15% of juxtamedullary nephron. Actually, in normal condition, mostly cortical nephron do their function, and this juxtamedullary nephron will work under stress condition. The main difference between these two is that loop of Henle of cortical nephron ends in upper medullary region and just below the juxtamedullary region. However, the juxtamedullary nephron glomerulus is present above the, just at the later end part of cortex, and this loop of Henle continues till the inner zone of medulla. Okay, and they actually participate in osmoregulation, salt concentration regulation. These are just schematic diagram from the tubular system. This is cortical nephron, this is juxtamedullary nephron, and these are vascular system, vascular blood supply to the system. Just to remember these two scientists, there are more because that more studies are going on, and if you will we will not cover it here, but the abnormal condition of kidney, if we will talk, we will find that lots of scientists are actually working on this, but on the background basis that Marcello Marfisi, he is a famous scientist who actually discovered this pulmonary capillaries and alveoli, and he also studied the detailed renal glomerular detail structure, and so under his name renal pyramids are called Malpisi pyramids. And this Sir William Bowman's, he's the one who discovered this Bowman's capsule, this cup like sac, and under his name only it's called Bowman's capsule. Now comes the renal circulation. As we know that the kidney's main function is purification of blood, primary function, if we will say, it's purification of blood, and purification of blood in one way, and another is we can say that water balance or electrolyte balance. So when we talk about water balance, electrolyte balance, we, from where we will get it, we talk about extracellular fluid, intracellular fluid, or plasma. So whatever the concentration of intracellular fluid or extracellular fluid, who contributes on it? Actually, blood contributes on it. So wherever there is an abnormality or the change in concentration of those two fluids of our body, that blood has to play a role or has to uh, participate in adjustment, and that blood does blood blood does actually, the kidney does, and it's through blood. So mostly that renal circulation is very important, is just to remind you that kidney makes only 1% of body mass. Okay, however, it gets 25% of cardiac output. supply of blood is 25% of cardiac output. And so the supply, if we'll see the supply of blood, this is the renal artery, this is renal vein. Renal artery then divides into segmental arteries, and that again form the interlobular artery, then arcuate artery actually, arcuate artery, and then from arcuate artery only your afferent arterioles starts, and then it collects into efferent arterioles and efferent arterioles, and then it will peritubular capillary, and then it will come into vein, and then arcuate vein, and then again it will go to interlobular vein, and then finally renal vein. This renal vein, especially the renal artery, is very thick and short, and that is the one reason that it can induce a high pressure on this blood so that filtration will happen, that I will come into that detail. And this is just for one slide to show in whole kidney. And if we will see on indivisible nephron, you will find that this is your afferent arterioles, and this is your efferent arterioles. Okay, and then they form the peritubular capillary for reabsorption of the components which is essential and came into the glomerular filter, that has to be again come into the blood. So that is happening through this peritubular capillaries, and then when they come, they form venules, and then interlobular vessels, and then arcuate vessels, and then renal artery. This previous also I have not discussed, but you have you saw that word vasa recta. Vasa recta is found in juxtamedullary nephrons, where this loop of Henle comes down to the medulla, and here there is this the capillary which come out from this afferent arterioles will come parallel to anti-clockwise parallel to this your loop of Henle, and these capillaries are called vasa recta. Okay, and they actually play a role in regulating the osmolarity regulation. Peculiarities of renal circulation is that renal circulation is a portal system, because here is one capillary and it just collects in efferent arterioles, and then it forms another capillary system. So it's a portal system, and the blood pressure is comparatively high, is about 60 to 75 millimeter of mercury, because this renal artery is short and wide, and the rate of blood supply in kidney is high, is 25%, and two circulation is happening. One is greater, that is on cortical nephron, and another is lesser, is on 15% on juxtamedullary nephron. Here another peculiarity I have not written, but I would like to mention that in cortical nephron, this the thickness of this afferent arterioles is more than efferent arterioles. The purpose is that this will create a good pressure on this glomerular tuft, so that more and more blood will get filtered through this glomerulus. Okay, so that is one another peculiarity actually. And the thing is that, as we have told, that this um, 60 mm or 75 mm mercury blood pressure is there in your renal artery, and so the pressure is here also, and that pressure is very important. This shaft is, you can think of that there is more than 90 or 1.2 lakhs nephron is there. So the individually glomerulus is very important. So if the blood pressure, like for example, if we know that normal blood pressure is 120 by 80 mmHg, and if for some person, if it will increase to more than 180 or 200 by 120 mmHg, so it will create a great effect on this. But nature wise, renal system, renal blood flow is auto-regulating. So this is till a physiological limit. Kidney has the peculiarity of maintaining the normal blood flow under any condition, and these are through this myogenic theory, and this glomerular tubular feedback mechanism. These two mechanism I will come later on. Just I mentioned here the name, and now I came into the this slide will deal with that what is the renal function. That we know that the primary function we know that formation of urine. We just say formation of urine, but through this process also the kidney plays an important role in water regulation or osmotic regulation, isn't it? So how it does, what are the process which is involved here is filtration. What part is involved? It is the woman's capsule play an important role, and we also call this not only filtration, we call this ultrafiltration also. And the glomerular capillary blood plays an important role. And then again, next comes the reabsorption. Total renal tubules and the peritubular capillaries, they play a role. And also the secretion, that is addition of certain solutes to the fluid in the tubules from the peritubular capillaries. Okay, so these three are main processes for formation of urine: filtration or ultrafiltration, reabsorption, and secretion. Okay, and finally excretion of extra water containing some solutes which we call urine. So schematically, if we will see that this blood comes through these efferent arterioles. Okay, this is glomerular capillary, and while passing through it, there almost 600 ml blood pass each time, and 125 ml get filtered here, 500 ml blood pass through here, and this get so first is ultrafiltration. Next, when it passed through the tubules, here one thing I would like to mention that mostly in one nephron, the whole starting from here, that initial state of proximal convoluted tubule till the collecting duct, the whole size of the tubule is almost 45 to 65 mm. So you can think of that this small nephron is actually so long, 45 to 60 mm. So this filtrate pass through these tubules. So second is when they pass through it, there is reabsorption occur, and through these peritubular capillaries, sometimes secretion occurs, and finally urinary excretion. So these are three, and finally the outcome is this excretion processes is going on in nephron. So just to let you think that when you just sieve the tea, you don't have to think anything. You just put the sieve on cup and you pour your whatever, maybe tea, and it will get filtered. But if you this is the same thing, but if you will think in physiological condition, because we have a closed system, in that case, that is sieving of tea, you have the atmospheric pressure 760 mmHg. But in physiological condition, in our case, we are not having that pressure. That physiological pressure is not, atmospheric pressure is not playing, especially in kidney. However, in lungs, we know that that negative pressure plays a role, and lungs, that I'm not going into it. But here, the filtration is only possible if the blood which is coming here will have a good pressure for filtration. If it can be create a pressure over here so that it will get filtered. So, if we will see that what is the net effective filtration pressure, who is contributing and who is opposing this filtration, then we'll see that actually the sum of hydrostatic and colloidal osmotic forces across glomerular membrane, these are and the forces either these forces either favor or oppose the filtration. So the net effective pressure is forces which promote filtration minus the forces which opposing this filtration. So what are they? This is partial pressure, osmotic pressure in and glomerular part. Okay, and the one which is favoring is that pressure which is in tubular part, Bowman's capsule part, that pressure. Okay, and another is the osmotic pressure. Sorry, this is the osmotic pressure in Bowman's capsule part, and this is the hydrostatic pressure in Bowman's capsule part plus osmotic pressure in glomerular part. Okay, so these are the actually they favor and they just oppose. So now come into the calculation. The hydrostatic pressure in glomerular capillary is 60 mmHg. We have seen that. We have also talked about it that the blood pressure which is coming in renal artery and then in your afferent artery also is 60 mmHg. So this is actually the one of the important determinants of glomerular filtration rate. Next, the Bowman's capsule colloidal osmotic pressure is 0. Okay, because we when as the get filtered, it just passed on through here. So there is not much colloidal pressure is there to oppose it. Sorry. Next is hydrostatic pressure in Bowman's capsule is 18 mmHg. Hydrostatic pressure, whatever water is there in your woman's capsule, this is opposing it. It is 18 mmHg. And glomerular capillary colloidal pressure is 32 mmHg. Okay, so now it's come is this is 60 plus 0 is not much, and then 18 plus 32, and finally it comes to 10 mmHg. That means the filtration which is happening, final net filtration pressure which is exerted here is 10 mmHg. Now, once filtration is over, the filtrate will pass through the tubules, and while passing through, as I have already shown you the epithelial cells, that the epithelial cells part will serve their different function. So in short or in summary, if we will see that reabsorption happens either through this paracellular pathway by diffusion, transcellular pathway by diffusion, or through active transport and also the passive diffusion. Okay, so if we consider that what are the process, as I've told that this is either through active transport, ATP dependent in this lumen part or in this um, peritubular part. Lumen, this is brush border cells. These are actually proximal convoluted tubal cells, either through active transport or through passive transport also. And the sodium and glucose and amino acid, which is almost in normal physiological condition, 100% get reabsorbed. And we know in GI tract also, gastrointestinal tube also, this glucose and amino acid transport is through secondary active transport. So here also the secondary active transport happens, that sodium potassium ATPase dependent transport is happening, and through this energy is used by this glucose and sodium and amino acid and sodium for reabsorption in the interstitial fluid and then into the blood. And here it's potassium and hydrogen ion transport is happening through counter transport also. And when you will see, just this is just one example to show that glucose filtered load and plasma glucose concentration for each and every, like amino acids or glucose or sodium or potassium, there is a threshold limit. Okay, till one concentration that kidney or kidney renal tubules, epithelial cells of renal tubule will reabsorb most of them. Since glucose and amino acid are 100% reabsorbed, so the example is given with this, that in normal condition, that most of the glucose will get 100% will get reabsorbed up to a certain limit. They will not come into filtrate. Okay, but if plasma glucose concentration is above this threshold limit, that is more than 200 milligram per 100 ml of blood, then they will start coming into urine. Okay. So there is a threshold limit for everything. Here is the, just to show you the in normal condition, that threshold limit, like glucose, if till 180 mmHg, millimeter, sorry, millimeter per 100 ml of blood, 100% got reabsorbed. Bicarbonate almost 99.9%, sodium 99.4%, and urea 50%, and creatinine all will come into your urine. So this is just to show you in normal condition how reabsorption is happening. Now, individual cells, they are taking part in different process of reabsorption. So in proximal convoluted tubule, almost 65% reabsorption is happens for sodium ion, chloride ion, bicarbonate ion, potassium ion, water, glucose, amino acids. Almost most of them got reabsorbed, and few hydrogen ions, organic acids, and bases especially, they got they got secreted in lumen. Okay. In thin descending loop of Henle, this thin descending loop of Henle is permeable to water mostly, but not any salt. So only reabsorption of water happens here. And thick ascending loop of Henle is totally impermeable to water. Only salts get reabsorbed. Sodium, chloride, potassium, calcium, bicarbonate, magnesium, like they get reabsorbed. And secretion of hydrogen ion is happening. Here in these cells also, you have to remember like this, you have to concentrate on hydrogen ion also, because Dr. Bharti in next presentation, she will talk on um, this pH hydrogen ion concentration related role of kidney on pH regulation, and the role of hydrogen ion and acid base balance actually. So in here are the some of the slides which will show you that whatever we talked about, that active transport and co-transport, there are some inhibitors of these pathways have been found, that if it's being treated with this, they get inhibited. So that proves that inhibitor actually proves that these processes are going on these tubules. Distal convoluted tubule in normal physiological condition, they are they reabsorb the salts mostly, sodium, chloride, calcium, and magnesium, and same is true for collecting ducts, sodium, chloride, bicarbonate, potassium, and hydrogen ion secretion. Only water reabsorption is happening when there is hormone called antidiuretic hormone comes into play. If time permits, I will talk on uh, that. I don't know whether I will have much time. Here also this active transport will get inhibited by thiazidine diuretics, and similar there are some sodium ion blocker, aldosterone antagonist. They just inhibit this pathway, the sodium potassium reabsorption in distal convoluted tubules. Now, just very important to give you a overview of how this glomerular filtration rate. Glomerular filtration rate is the amount of filtrate form per minute. Okay, and they have very good relation with the, they have to be have very good relation with the renal blood flow. More renal blood flow will be there more glomerular filtration will be there. But if you will find see this graph, you will see that glomerular blood flow is also regulated, and parallelly glomerular filtration rate is also regulated. And here if you will see that arterial blood pressure from 50 to 200 mmHg increased. However, that urine output is not increased because they are regulated. Okay. So there are some of the factors which are actually affecting glomerular filtration rate. They are changes in renal blood flow, changes in glomerular capillary hydrostatic pressure, systemic blood pressure, efferent arteriolar constriction, hydrostatic pressure. I will just pass on three because these are important, some more important parts to be mentioned that will not cover the time. This countercurrent multiplier system is active in Henle's loop of juxtamedullary nephron, where the concentration, this is descending loop of Henle. We have seen that the descending loop is permeable to water but not salts. So most of the water comes out, and the filtrate which is coming down is now osmolarity is very high, and when it is going up, as this part is impermeable to water, so most of the salt is coming out, and now this glomerular filtrate is coming into normal as its normal condition. Okay, and the interstitial fluid concentration is maintained. So this is countercurrent mechanism, multiplier system. Actually, the individual processes will take a lots of time. Individual classes, I will just pass on things because this is all one of the important thing to mention that that is actually play important role in autoregulation. This is the structure of juxtaglomerular apparatus. Juxta means close to glomerulus. Okay, this is the structure where the distal convoluted tubule of the same nephron, when it passes through close to glomerulus, it forms an important structure called juxtaglomerular apparatus, and it has some modified cells also. These efferent arterioles and part of efferent arterioles, they have juxtaglomerular cells, and this distal convoluted tubules has macula densa cells, and there are vascular smooth muscle fibers is there. Here the Lassie cells is there, and some mesangial cells is also there. So these structure actually play a very important role in um, controlling the glomerular filtration rate. This is structure. This is the glomerulus. Okay, these are efferent arterioles, efferent arterioles. These are JG cells and macula densa cells. So actually, what happens when vascular smooth muscle cells is also there, and this mesangial cells is also there. Actually, just to mention that actually 5 different type of cells is there: juxtaglomerular cells, vascular smooth muscle cells, macula densa cells, Lassie cells, and mesangial cells. Okay, when pressure increased in these afferent arterioles, they get sensed by these extraglomerular cells. These extraglomerular cells actually contain the renin. Renin now comes into blood, and it activates angiotensinogen into angiotensin 1, and angiotensin 1 in the lungs, I think it's in the next slide, it converted into angiotensin 2, and this angiotensin 2 has vasoconstriction effect, and by vasoconstriction it regulate the pressure exerted on this wall of glomerulus. And another regulation is that when the filtrate pass through this macula densa cells, if salt concentration is high, it's sensed by your macula densa cells, and this cause the release of aldosterone, and that way also is regulated. I will just come if possible. This is the myogenic effect, as I have told in a previous slide. The regulation is done by myogenic response and tubular glomerular feedback. Myogenic response is efferent arterioles, pressure is increased, sensed by JG cells, and now JG cells will, after release of renin, it will cause vasoconstriction, and so pressure is regulated. Okay, and through tubuloglomerular feedback, when the filtrate pass through this distal convoluted tubule, that salt concentration is sensed by these macula densa cells, and that cause the release of aldosterone from your adrenal cortex, and that cause reabsorption of salts from your filtrate, and that way it's cause the vasodilation, vasoconstriction also different way. So the function of JGAs is secretion of two important hormones, renin and prostaglandins, and by the way, it regulates the renal blood flow and glomerular filtration rate, and it also show the response with elevated sodium through macula densa, and thus it reduced the blood flow to the glomerulus, and the glomerular filtration rate is also reduced. And these JG cells also secrete renin when blood pressure is high, as I have already discussed it, and I am not going to discuss it. This is just, I will like to just mention that this renin released from kidney, and this angiotensinogen is synthesized in liver. Renin cause angiotensinogen to convert into angiotensin 1. This is an decapeptide, and angiotensinogen converting enzyme convert it in liver into angiotensin 2, and this cause vasoconstriction as well as release of aldosterone from adrenal cortex, and this cause stimulates the sodium ion and water reabsorption in the nephron. Okay. So now finally, let me come into the process of micturition, that is your release of once the urine come into renal pelvis through peristaltic movement, it comes into your urinary bladder. This urinary bladder is made of mostly detrusor muscle, which has both circular and longitudinal smooth muscle cells, and they have nerve fiber, both sympathetic and parasympathetic, as well as pudendal. This pudendal nerve sulci is through its skeletal muscle, so its external sphincter is under our control till a point. However, this sympathetic supply is called relaxation of this detrusal muscle, so that more and more urine will get collected here. And this parasympathetic stimulation cause the constriction of this detrusal muscle, and so the release of urine through urethra. This whole micturition process also needs very detailing study, that maybe individually we will discuss in some other class. And this normal this the pressure on this urinary bladder is depends on this volume. The volume which is can be stored in urinary bladder is almost 300 to 400 ml, and from 200 ml urine, as soon as 200 ml urine collected in your urinary bladder, you can feel the pressure, and that pressure will keep on increasing as much as urine is being collected in your urinary bladder. And after more than 300 ml, it's almost strong, no more increase in fluctuation. This is the limit, and that can be stand for some time. Otherwise, you have to release the urine in your system from the urethra. So I think just just to mention the function that kidney actually primary function is regulation of homeostasis, that is the concentration of electrolytes, mostly sodium, potassium, and chloride, and water concentration also, acid base balance, and minerals also is regulated, and the waste material, removal of waste material like urea, creatine, and uric acid. And the important is that the kidney has the endocrine function. It releases erythropoietin in the hypoxia condition. As you have seen that you have a lots of mitochondria in epithelial cells of your tubules, so they require oxygen. So if anyone have less um, hemoglobin concentration in their blood, so they will the tubules will feel that hypoxic situation, and that will they will cause, and the erythropoietin will get released from cells of kidney, and they will affect on stem cell erythrocyte stem cells, and cause more and more erythrocyte synthesis. And one important hormone is 125 dihydroxycholecalciferol, that is the precursor of vitamin D3. Vitamin D synthesis, renin, one of the important hormone which for required for vasoconstriction. In addition to that, bradykinin, prostaglandin, and endothelial factors, and as well as nitric oxide and endothelin are also released. The secondary function, if we will call it, is just filtration, reabsorption, and secretion. That is for formation of urine. And they also regulate secondary function. They does like regulation of blood pressure through renin, red blood cell production through erythropoietin, or calcium absorption by conversion of calcidiol into calcitriol. Okay. If anyone is there, I think I should not continue much because already almost a year hour is gone. So these are the some of the kidney function for measuring the function of kidney, that is clearance rate, glomerular filtration rate, and clearance ratio, effective renal plasma flow, like this. So few slides are there, just their definition. So definition students can just read from here. Maybe in some other class we will have more elaboration on all of them. And just to show you a schematic diagram, that those who have that kidney failure, they have to go through that dialysis, because the continuous purification of your blood is necessary to keep the homeostasis under control, to to keep your internal media of your body, especially pH, electrolyte concentration, and water concentration in your body. So most of the information which I gave you is from these two textbooks, Ganong and Brighton textbook, Medical Physiology and a Review of Medical Physiology. And most of the pictures are from Common Creative License and some from these books. Okay, thank you very much. These are the my two email ID. So students can contact me if they will have questions for from today's presentation. So thank you. Let me stop my screen sharing.
Thank you, Dr. Raki. You did a great job. Nearly 10 hours content.
Right.
You
In one
could
hour.
you could concentrate and deliver it within less than 1 hour time. So it's a great job.
Actually,
And
we individual thing we will elaborate maybe in individual class, isn't it? If required.
surely this was planned for only overview of the excretory system.
Right.
So you have done the right thing. For the viewers, those who are attending or those who will be attending later, I can suggest that uh, see Dr. Raki is before joining as assistant professor in Belda College, she had a very colorful research career. She stood first, university ranked university first from Vidyasagar University, where from I am also belonging from that same university. And then she did MSc from Calcutta University, and then PhD in molecular biology from Benares Hindu University. And then Dr. Raki had many research fellowships, including UGC, DBT, NIH from US, and postdoctoral fellowships. Along with that, she has also served as a scientist in NICED, Calcutta, and also as BioCare DBT women scientist. So after from those level to come back to UG level and concentrating these information in a very lucid but concentrated way, it's really tough. I congratulate you, and I can see you are not very well also. Physically also you had some problem. That's still I really appreciate you have continued, and it's really nice. And Sinchan Mukherjee as mentioned that it was nice presentation. Very few are online today. Today is a festive night.
Right, right.
Even then, you have spared your time to become live in this session. I really appreciate and I thank you from the on behalf of Education Committee, PSI Education Committee, from the PSI as well as from the audience. I thank you, appreciate your effort. Thank you very much. Thank you very much.
Thank
And uh, if possible, we can share your email ID in
Yes,
the chat so that you can join that YouTube chat and you can share your email ID so that anyone who wants to communicate to you directly can communicate to you.
yes.
Thank you very much, and thank you. Our next session we will be having another great physiologist, Professor Bharati Mehta. She is currently working as professor in the Department of Physiology, AIMS Jodhpur, and I am also from there. I am also working with Madam. It's my privilege. She has completed MD in physiology as well as she is PhD in physiology. So it's a rare combination, MD, PhD, rare combination. She has around 23 years of teaching experience, teaching physiology in various UG as well as PG courses. She has written a book, published a book titled MCQs in Medical Physiology. Those who are interested, they can enjoy that book. She is Her research area is respiratory physiology, sleep medicine, as well as medical education. So she is really concentrating on teaching physiology for the undergraduate as well as postgraduate, and she has a good name. I know. Being her colleague, I know she has a very good name among the students. So I believe students will be happy. And along with the teaching in physiology, she is currently working in a multicentric ICMR project and a WHO project. She is also conferred with FAMR Fellowship, which is a fellowship for the medical education, and she is also has the membership of National Academy of Medical Sciences. She is also member of editorial board and reviewer panel for many national and international journals. And I think, Madam, welcome you all. And Madam, I welcome you also in the session.
Thank you so much. Thank you.
And now, with the background work Dr. Rakhi has given, so I think it's better we continue, and Madam's presentation will be shortly starting. In the meantime, we can have Dr. Rakhi email ID is available in the chat. So those who are interested, they can send their communication to Dr. Rakhi. And I think later we will give Dr. Mehta Madam's email ID also will be sharing.
Sure.
So not much waiting, Madam. Stage is
Thank
yours.
thank you so much. And a very good evening to all of you. Uh, First of all, thank you, Dr. Nayak, for introducing me to the house. And I also thank the Physiological Society of India for giving me this opportunity to present on this prestigious platform. Uh, Education Committee is doing a great job, and this initiation is definitely going to help a lot of students. I congratulate the Education Committee of PSI for that. And I also thank Dr. Rakhi, the previous presenter, who has set the stage, who has touched all the points where I'll be going. And so it is like she has presented the abstract, and now I'll be presenting the full paper. So thank you, and I'll just share my screen. Uh, is my screen visible?
Yes, ma'am.
Is it visible?
Yes, ma'am. Your screens are visible.
Okay, thank you so much.
Yeah.
So I will be talking about defense of hydrogen ions by the kidneys, commonly known as acid-base balance. Now, hydrogen ion concentration in ECF is 0.00004 milliequivalent per liter. So, with such a small concentration of hydrogen ions in the extracellular fluid, even then the hydrogen ions are very important to maintain all the biochemical reactions which are responsible, which are crucial for the homeostasis to take place. And therefore the body needs to regulate the hydrogen ions very, very meticulously and in a very, very narrow limit. The hydrogen ion concentration is expressed as pH. P actually comes from a German word. Interestingly, when I was seeing, it means potens, and H with it, potens means power. And so pH means the power of hydrogen ion, and it really is a big power of hydrogen ion. We will just see that it has got so many, it can affect the bodily functions very adversely if the range is disturbed. So, since it has got such a crucial work to do, so it is not surprising that more than one systems in the body are responsible to maintain its concentration to a normal level, and which is a very low, very narrow range, 7.36 to 7.44. Before delving deep into the regulation of hydrogen ions, let us quickly recap some of the definitions which we already know. Acid is a molecule that releases hydrogen ion in the solution, and the base is the one which accepts it. The stronger the acid, the more rapidly or more readily it will release the hydrogen ions. The example being hydrochloric acid, sulfuric acid. And weak acids also release hydrogen ions, but they do it very slowly. And the example is carbonic acid. Similarly, the strong bases are ones which rapidly accept the hydrogen ions, and weak ones are those which accept the hydrogen ions but slowly. Buffers are weak acids or bases along with their conjugate salts. Now, as I told you that hydrogen ion is very important, it has to be regulated in a very narrow range. There are three systems in the body which are regulating the hydrogen ion concentration in the blood. First of all, and the first to occur, is the buffer systems of the body. So there are a number of buffer systems in the body. Some are there in the extracellular fluid, some are there in the intracellular fluid, and all of them have got the function of buffering the hydrogen ion concentration. Like if the hydrogen ion increases, they will immediately accept those hydrogen ions so that the pH of the system or pH of the fluid does not change much. And they are the first to act, they are the quickest of them, quickest of all the regulating mechanisms. And during the process of buffering, they are consumed up, and the body will have a system to replenish these buffers, which we will just see, and that is the function of kidney. Another system which is meant to regulate the hydrogen ions concentration in the body is the respiratory center, and this acts by changing the ventilation, Ventilation will change the carbon dioxide concentration in the blood, and carbon dioxide is a potential acid. Why? Because carbon dioxide readily combines with water to form carbonic acid. So, carbon dioxide is considered to be a potential acid, and by changing the ventilation, the respiration can either excrete out more carbon dioxide, expire out more carbon dioxide, or it can retain carbon dioxide depending upon the pH of the body. The kidneys and the respiratory center acts in hours, in minutes to hours. The kidneys are the mainstay of uh, maintaining the acid base balance, and they do so by either excreting the acidic urine or alkaline urine, and they maintain the hydrogen ion concentration. With this, it may take around 2 to 3 days for the kidneys to come to its fullest potential, but this is the strongest mechanism to maintain the acid base balance of the body. Buffers do not eliminate or add hydrogen ions to the body. They just keep them tied up, and they do not change and they don't let the pH change much until the balance is re-established by respiratory center and kidneys. So buffers are the temporary mechanism, makeshift arrangement to keep the hydrogen ion concentration in a uh, an acceptable range without changing the pH much. Now, in the body, there is daily production of acids, and these acids are produced because of the metabolic processes in the body. So, we have metabolic processes going on, and which generate acids, and the acids which are produced in the body are classified as two types. One is respiratory or volatile acids, amounting to 12,000 to 13,000 millimoles per day. Now, as the name suggests, volatile acids means which are in the air form. And what is the acid which will be in the air form? It is the carbon dioxide. So the carbon dioxide that is generated in the cells, we know that lungs uh, take in oxygen and they give out carbon dioxide. So it is this carbon dioxide which is of the amount which amounts to 12,000 to 13,000 millimoles per day, and which is excreted out by the lungs every day. Another form of acids that body produces is the metabolic or the fixed acids. These are the acids which are other than carbon dioxide and carbonic acid, because those are the volatile acids. So these are other acids which cannot be expired out, or which cannot be given out or excreted out by the respiratory system, although they are smaller in amount, but kidney is the sole source for excretion of these fixed acids or the metabolic acids. Now, volatile acids, as I told you, are carbon dioxide and carbonic acid. And carbon dioxide is formed by the complete oxidation of carbohydrates and fatty acids. So when the nutrients are completely oxidized, they are converted into carbon dioxide and water, and carbon dioxide is expired out through the lungs. So basal carbon dioxide production is 12,000 to 13,000 millimoles, and which is excreted out by the lungs. The fixed acids, on the other hand, are all those acids other than other than carbonic acid, and they are those acids which cannot be excreted out by the lungs. They are produced due to incomplete metabolism of the nutrients, like lactic acids, ketone bodies, sulfates, phosphates. So all these are produced by incomplete metabolism of nutrients, and these fixed acids, the net production of which is 70 to 100 millimoles of hydrogen ions per day, and these are excreted only by the kidney. This is non-volatile acid load. Now, what will be the body's response to hydrogen ions? So, we must understand that in health, a normal person generates this much amount of acid daily in the body, and the kidneys are supposed to excrete out all the fixed acid, and the respiratory system is meant to uh, release all the volatile acid. But suppose if the respiratory system is not functioning normally, that can also lead to increased acidity or alkalinity. And in that case, the kidneys will take the function, the kidneys will uh, try to excrete out all the hydrogen ions which are present in the body. Okay. So the body's response to hydrogen ion is, first defense will be buffering, which acts immediately. Second defense is by the respiratory system by altering the ventilation and eventually the arterial PCO2, which happens in minutes to hours. And the third defense mechanism will be alteration in the bicarbonate and hydrogen ion excretion. So kidneys have a provision to either excrete the bicarbonate or reabsorb the bicarbonate, excrete the hydrogen ions depending upon the pH of the body. Normally, because every day normally the acid is generated and that has to be excreted out of the body. So the kidney has to conserve all the bicarbonate and has to excrete some hydrogen ions. We will just see. Now, what happens when the acid is entering into the blood? When the acid is produced into the blood and it is entered into the blood, buffering happens immediately. And this is how the buffering happens. First extracellular buffer will be the bicarbonate buffer to act. So the equation or the reaction will be bicarbonate combining with the hydrogen ion to form carbon dioxide and water, and this carbon dioxide is expired through the lungs. So this is normally that happens. And besides bicarbonate buffer, there are some non-bicarbonate buffers also available in the blood. And a general equation can be written as B stands for buffer, combining with hydrogen ion to form BH. So this happens immediately when the hydrogen ion enters into the blood, buffering, first thing to happen. And in this process, the bicarbonates are consumed because carbon dioxide and water is formed and carbon dioxide is excreted out. So bicarbonates are consumed and non-bicarbonate buffers are also decreased in the blood. So now body has got a function, a job, a task to replenish those buffers which are used up during buffering of the hydrogen ions. Next come into play the respiration. Now the acid, this happens is in within minutes to hours, and the respiratory system causes changes in pH by alteration in the ventilation, which changes the PCO2 levels in the blood, and CO2 is the potential acid. So normally when the acid is generated in the body, this hydrogen ion, we know that hydrogen ions will stimulate the respiratory center, and this will increase the ventilation. So when the ventilation increases, the PCO2 level is washed out. So increased oxygen entering into the body and excessive carbon dioxide going out of the body. So the respiratory compensation for the hydrogen ion entering into the blood will be hyperventilation and leading to carbon dioxide excretion. And vice versa is also true, that if there is hypoventilation or if there is alkalosis, so hydrogen ions are less in number, less in amount, the hydrogen ion concentration is less. In that case, the ventilation will be suppressed, and therefore this will lead to carbon dioxide accumulation in the blood and leading to acidity. So because in alkalosis we want acid to be accumulated so that the alkalosis is corrected. So this is what happens at the level of the respiratory system. And once the carbon dioxide levels are changed, carbon dioxide is a very lipid soluble gas. It quickly crosses all the cell membranes, and so the changes in the PCO2 result in rapid changes in the hydrogen ion concentration in all the fluid compartments of the body. So this is how the respiration acts, like respiration acting to control the hydrogen ion concentration. Now, how the kidney functions. Now, kidney is left with three functions. One is that if the kidney is to kidney faces an acid load, in that case, the kidney must excrete that acid. Number one. Number two, it should reabsorb all the bicarbonate which is filtered, because bicarbonate is an alkali, and loss of alkali in the glomerular filtrate leads to is equivalent to an acid load to the body. So the bicarbonate has to be reabsorbed completely, because we know that bicarbonate is present in the blood and it is filtered through the glomeruli, and if it is not reabsorbed by the kidney back, it will lead to more acidosis. So the kidney must excrete the fixed acid which is produced in the body. It must reabsorb all the bicarbonate which is filtered in the glomerular filtrate. And the third function is that it should replenish the buffers which are used up once the acid load was there in the blood. So, how does kidney do this? The kidneys remove all the daily production of non-volatile acids, and to maintain the acid base balance, the kidneys must not only excrete this fixed load of hydrogen ion, it must also reabsorb virtually all the filtered bicarbonate. Both these processes require hydrogen ion secretion into the lumen. Now, for bicarbonate reabsorption also, the kidney needs to secrete hydrogen ions in the lumen. And for excretion of fixed acids, again, the kidney has to secrete the hydrogen ions into the lumen. And the total amount of secretion of hydrogen ions which is required for reabsorption of bicarbonate is far greater than that required for excretion of the fixed acid. 4,000 to 5,000 millimoles of hydrogen ions are secreted per day for just reabsorption of bicarbonate, and 70 millimoles of hydrogen ions are secreted per day for excretion of the fixed acid which is developed in the body per day. And where is this happening? Okay, one more thing which is very important to note is that secretion of every hydrogen ion will generate and reabsorb one bicarbonate ion. So every time a hydrogen ion is generated or secreted by the kidney, one bicarbonate ion is formed and that is reabsorbed into the blood. And we will see how it is happening and what are the functions of this bicarbonate which is formed. Now, secretion of hydrogen ion, where does it take place? Now, kidney has to secrete hydrogen ions for bicarbonate reabsorption, and it has to secrete hydrogen ions for hydrogen ion excretion. Okay, so for reabsorption of the secretion of hydrogen ions in the kidneys take place at two places. One is the proximal convoluted tubule, and another is the intercalated cells of distal convoluted tubule and collecting tubules. So at these two places only, the secretion of hydrogen ion takes place. In between, that is in the loop of Henle, there is no such secretion taking place. Now, at the PCT, the hydrogen ion secretion is used for bicarbonate reabsorption. Remember, the kidney needs to reabsorb all the bicarbonate which is filtered. Okay, and this reabsorption, 90 to 95% of the bicarbonate reabsorption takes place at the proximal convoluted tubule. And now, once the bicarbonate is reabsorbed, now the kidney is left with excretion of the fixed acid, 70 to 100 millimoles. That takes place at the level of the DCT and collecting ducts. So let us see. And when the bicarbonate reabsorption, when the hydrogen ion secretion takes place at the level of the proximal convoluted tubule, this hydrogen is used up for bicarbonate reabsorption, and therefore there is no net loss of hydrogen ion. But in the DCT and CT, when the hydrogen ion secretion takes place, it causes a net loss of hydrogen ion, which is required because we want to lose hydrogen ions which are generated in the body daily. So, how the kidney reabsorbs the uh, bicarbonate ions at the PCT? Now, normally the glomerular filtrate is about 180 liters of blood plasma. 180 liters. It is a huge amount. And we know because 120 ml per minute is the GFR, and this amounts to 180 liters of blood plasma per day, filtering per day. Each liter contains 24 millimoles of bicarbonate, and therefore 180 multiplied by 24, this is 4320 millimoles of bicarbonate which are filtered. So if all this filtered bicarbonate is not reabsorbed, then what will happen? This would be equivalent to an acid load of 4320 millimoles. So our body could not afford this. So kidney reabsorbs all the filtered bicarbonate. And how does it do so? In the proximal convoluted tubule. So this is a proximal convoluted tubular cell. This is the blood, and here is the lumen. So reabsorption will take place here in the blood, and secretion or excretion will take place in the lumen. So what happens in the PCT cell? In the PCT cell, there will be at the basolateral membrane sodium potassium pump, and in the PCT cell, the carbon dioxide will combine with water to form carbonic acid. Normally, this process is very slow, but PCT cells are rich in an enzyme called carbonic anhydrase, which speeds up this reaction many times, many folds. So, when carbonic acid is formed, it immediately dissociates to form hydrogen ion and bicarbonate ions. These hydrogen ions are they have to be released into the lumen. Okay, and when for releasing into the lumen, they require a transporter, and here the transporter is sodium hydrogen antiport. This was shown to you by Dr. Rakhi as well. So, this sodium hydrogen antiport means it moves the sodium in and hydrogen out. Now, this transporter requires sodium, and the gradient for sodium is maintained by sodium potassium pump. Now, this hydrogen ion, when it enters into the lumen, it will immediately combine with bicarbonate, and this bicarbonate will again be converted into carbonic acid. This hydrogen plus bicarbonate will be converted into carbonic acid, and this will be dissociating into carbon dioxide and water in the presence of carbonic anhydrase. So that means the carbonic anhydrase is present not only inside the PCT cell, but it is also present on the brush border of the PCT cell, which is there in the lumen. So effectively, carbonic anhydrase is present in the lumen for this reaction to speed up. Okay, and what happens? This carbon dioxide it enters into the PCT cell, and it is available for this reaction to take place. So this is the way in which the hydrogen ion is secreted into the lumen. Now, where is this bicarbonate coming from? This bicarbonate is the filtered bicarbonate, which is there in the lumen, and we need to reabsorb it. What you are seeing here is that this bicarbonate has combined with this, and this has converted into carbon dioxide and water. That means this has been used up, but we need to reabsorb it. So what is happening? You must also remember that I told you that every hydrogen ion, if it is generated, a bicarbonate is also generated. and it is this bicarbonate which will enter into the blood. So this bicarbonate will be reabsorbed. So what you are seeing here is one bicarbonate is lost, the filtered bicarbonate, one is lost, but at the same time, one is reabsorbed. So this is how reabsorption of bicarbonate ions take place in the PCT. Although this bicarbonate and this bicarbonate is not the same molecule which is entering into the blood, but the number of molecules are same. That means in this process, this hydrogen ion is actually being buffered by this carbonate, and it is used up generating carbon dioxide, but in the process one bicarbonate is reabsorbed. So in effect, the number of filtered ions of bicarbonate are all reabsorbed in the blood in the proximal convoluted tubule. So, bicarbonate reabsorption, therefore, does not represent the net hydrogen ion excretion. We have seen that hydrogen ion was converted into carbon dioxide and water. Some of the carbon dioxide will be expired out, and some will be some is entering again into the PCT cell for the reaction to take place. So this is not the net excretion of hydrogen ions which is taking place. This is bicarbonate reabsorption that is taking place, and the hydrogen ion secretion is used for this. Although the amount of bicarbonate reabsorption is simply the reclamation effort, this process consumes the largest fraction of hydrogen ion secreted into the tubule lumen. So for reabsorbing 4320 millimoles of bicarbonate, 4320 millimoles of hydrogen ions were secreted into the PCT lumen, and this is such a huge amount of hydrogen ions. So, and this is done by sodium hydrogen antiporter. Now, once the bicarbonate is reabsorbed, the kidney is now left with the fixed acid which is to be excreted. And as none of this happen, none of the hydrogen ion secretion takes place in the loop of Henle, now the fluid will move to the intercalated cells of the distal convoluted tubule and the collecting ducts. Again, this is the intercalated cell, and how the hydrogen ion will be secreted, the process will be same. The same carbon dioxide, and this carbon dioxide will be combining with water to form carbonic acid, and with that will be dissociating to release one hydrogen ion, and one bicarbonate ion will be reabsorbed into the blood. But here the difference is the transporters. The transporters here are different. So intercalated cell also has got sodium potassium pump at the basolateral membrane, but the transporters here are sodium. It is not dependent on sodium, so no sodium transporter. It is the hydrogen, potassium hydrogen ATPase and hydrogen ATPase. So these are pumps which directly involve energy, utilize energy, and these can secrete the hydrogen ions against a very large gradient. So the reaction will be same: carbon dioxide plus water to form carbonic acid, and here intercalated cells are also rich in carbonic anhydrase. Carbonic acid converting into hydrogen ion and bicarbonate ion. This hydrogen ion will be excreted into the lumen either by this transporter or this pump, or by this pump. And this bicarbonate will be reabsorbed, and here it is an antiport which is called bicarbonate chloride antiport, and this chloride will enter into the cell, and this chloride from here will go into the lumen. So in effect, what we are seeing is that bicarbonate is reabsorbed, and this bicarbonate, we don't have we there is nothing to be reabsorbed now. So this bicarbonate which is generated is a new bicarbonate. Okay, the body is gaining one bicarbonate, and what will this bicarbonate do? This bicarbonate will be utilized to replenish the buffers. Remember, the kidney has got a third function: to replenish the buffers that were used up. So the kidney will replenish these buffers by with the help of this new bicarbonate. We will just see. And in effect, what is happening? Hydrogen and chloride is being released into the lumen to be excreted out. So the kidney has to excrete the fixed acid, and hydrochloric acid is the fixed acid, which will be excreted out in the urine. Now, how the kidney, so the new bicarbonate which was generated, I told you that this will be utilized to replenish the buffers. One is the carbonate buffer and one is the non-bicarbonate buffers. Okay, so now what will be the how will the bicarbonate generate the base B? This is the reaction. Bicarbonate will convert with this when the base buffered the hydrogen ions. This was the resultant product which was formed. So bicarbonate will combine with BH to form the base or the alkali or the buffer, and carbon dioxide and water, and again this carbon dioxide will be excreted out of the lungs. So what we have seen is that the kidney has reabsorbed all the bicarbonate which was filtered in the PCT. It has secreted the hydrogen ions in the lumen because it has to excrete the fixed acids, and it has replenished the buffers. So it has done all the three tasks, but the excretion of fixed acid has got, there is more to do to it. The normal excretion, normal amount of urine that is formed in the body is the normal amount of urine that is formed in the body is 1.5 liters. Okay, and how much fixed acid is to be excreted in the urine? It is 70 millimoles per liter. So if this fixed acid enters into the urine unbuffered, then what will happen? It will reduce the pH of the urine to say 1.6, and kidneys cannot achieve that. The minimum pH that the kidneys can achieve is just 4.4, and that is called the limiting pH. So how the kidneys tackle this problem? So this is again the buffers coming to play. Now the buffers were there in the blood. They were, what was the function of the buffers? They were not letting the pH to change much. Thing is needed here in the kidneys. We need buffers in the lumen. Now, what kind of buffers will be there? Now, what are the buffers? So buffers which this is the same thing which I have told. The buffers, where are these buffers coming from? So the buffers, some of the buffers are filtered. They are there in the glomerular filtrate, like phosphate buffer. And some of the buffers are there which the kidney can generate or kidney can synthesize. So if the kidney has to excrete more acid, it can synthesize more buffer. And if the kidney does not have acid to excrete, the kidney will not synthesize that buffer. So the kidney will now utilize those buffers. And how the buffering is taking place? The uh, again, it is the same thing. I should have changed the slides. If there were no buffers that tied up the hydrogen ions in the urine, the limiting pH would have reached rapidly. So the kidneys need to bind the hydrogen ion to the buffers in the lumen. Now, the kidney filters some of the buffers like phosphate buffer, creatinine buffer, urate buffer, and it synthesizes some buffers like the ammonia buffer. Now, phosphate is the most important filtered buffer. Now, why is it important here at the intercalated cells? Because bicarbonate has been reabsorbed. It is no longer present in the lumen, number one. And number two, Dr. Rakhi told you that in the proximal convoluted tubule, 65% of the water is reabsorbed. 65% of the filtrate is reabsorbed. Another 20% is reabsorbed in the loop of the Henle. So by the time the filtrate reaches the distal convoluted tubule, the phosphate buffer are concentrated. So they are available for combining with the hydrogen ions. And another thing is that the pK of the buffer, this phosphate buffer is 6.8, and the fluid when reaches here is almost of this pH at this. So the phosphate buffer is available readily for combining with hydrogen ions. But again, this is limited because this was present in the filtrate. Okay, and whatever hydrogen ions which are combining with phosphate, they add to the titrable acidity of the urine. So titrable acidity of the urine is given by the phosphate buffers. Now another, suppose the phosphate buffers, along with that, the kidney, what is the buffer that the kidney can synthesize? It is the ammonium buffer. So depending upon the need, the kidney can increase or decrease the synthesis of ammonia, because phosphate is limited. Phosphate cannot be increased. If the need is more, then the ammonia buffer can be synthesized. So ammonia reacts with secreted hydrogen ion to form NH4 ammonium ion, and the pK of this system is 9. So this does not contribute in the titrable acidity, because we titrate only up to 7.4, but the pK of this buffer is 9. So this buffer does not contribute to the titrable acidity of the urine. Titrable acidity is given by the phosphate buffer. And this, with this, we can understand that if the kidney needs to excrete more acid, for example, in respiratory acidosis or metabolic acidosis, in those cases, the phosphate buffer will increase in the urine, and the ammonia buffer will also increase in the urine. And opposite will be true in the alkalosis. In alkalosis, when the kidney do not require to excrete acid in the urine, then the phosphate buffer will also be low and the ammonia buffer will also be low. We will come to it and we'll talk about it quickly in last few slides. So the fate of hydrogen ion in the urine is, in the PCT, it was used to it combined with bicarbonate, and it was utilized for reabsorption of bicarbonate. And in the DCT, these two reactions took place. The same is here. So this is the three ways in which the hydrogen acts, or hydrogen ion reacts in the lumen. First is in the proximal convoluted tubule. When the hydrogen ion is released into the lumen, it combines with bicarbonate to form carbon dioxide and water. And remember, the bicarbonate will be reabsorbed. Every time a hydrogen ion is secreted into the lumen, a bicarbonate will be reabsorbed into the blood. So this bicarbonate, which is reabsorbed, is the reabsorbed bicarbonate, right? Like the filtered bicarbonate, which is reabsorbed, and this occurs at the level of the PCT. In the DCT, two processes can happen. One is that the hydrogen ion which is released will combine with the phosphate ion. This will give the titrable acidity to the urine, and this will generate the bicarbonate, because every time a hydrogen is secreted, a bicarbonate is reabsorbed. I am saying it again and again so that the students should never forget this. And when this bicarbonate is the new bicarbonate which is utilized to replenish the buffers, again the third fate of hydrogen ion will be combination with ammonium. And this ammonium this will be combination with ammonium, and the bicarbonate which is reabsorbed is, again, this is also a new bicarbonate which is utilized to replenish the buffers. So what are the pH changes that are happening in the nephrons? Now in the PCT, although 4320 millimoles of the hydrogen ions were secreted, but all of them were buffered by the bicarbonate, and they were utilized to reabsorb the bicarbonate ions. And therefore, the pH changed only to 6.8. So the capacity of secretion of hydrogen ions was great, but the gradient that changed was very less. So this system, proximal tubule, is a high capacity, low gradient system for hydrogen ion secretion. That means high capacity 4320 millimoles of of hydrogen ions were secreted, but the gradient changed only up to 6.4. So this is the proximal tubular system is a high capacity, low gradient system for hydrogen ion secretion. And in the DCT and collecting tubules, the secretion of hydrogen ions is just to excrete out the fixed acid, that means of the amount of say 70 to 100 millimoles, but the pH changes to 4.4. So this system is called low capacity high gradient system for hydrogen ion transport or hydrogen ion secretion. So this is the summary. The kidneys secrete 4390 millimoles of hydrogen ions per day into the tubular lumen. In health, we are talking, out of which 90%, 98% of which is utilized to filter, to reabsorb the filtered bicarbonate, and rest is utilized, or rest is the generation of new bicarbonate, which is utilized for uh, replenishment of the buffers. What are the differences in the hydrogen ion secretion at the level of PCT and at the level of intercalated cells? At the PCT, the transporter is sodium hydrogen antipode, which is sodium dependent. So this kind of hydrogen ion secretion is sodium dependent. This hydrogen secretion is used up for reabsorbing the bicarbonate. It secretes hydrogen ions against a small gradient because there is no pump here. So the gradient of secretion is small. Amount secreted is huge. The buffers used are bicarbonate, and this system is called high capacity low gradient system. At the level of the intercalated cells, the transporter is hydrogen potassium transporter or the hydrogen pump, and therefore it is sodium independent. It generates new bicarbonates because the filtered bicarbonates were already reabsorbed at the level of the PCT. It can secrete hydrogen ion against a large gradient because the pump will directly utilize the energy. The amount secreted here is 70 millimoles per liter. The buffers used are phosphate buffer and ammonium buffer, and this system is called the low capacity high gradient system. So the kidneys reabsorb all the bicarbonate by secreting hydrogen ions into the PCT lumen. They excrete the fixed acid by secreting the hydrogen ions into the DCT and CT lumen, and the kidney also replenishes the buffers. So this is about the normal functioning of the kidney, or normally how the kidney maintains the hydrogen ion regulation. But there are some acid-base disorders. We won't be going into the detail. I'll just be touching these just to give you a crux of this. So there can be respiratory acidosis, respiratory alkalosis. That means when the respiratory system is abnormal. So what will happen if the respiratory system is abnormal? There will be either accumulation of carbon dioxide or washout of carbon dioxide. So accumulation of carbon dioxide, if it happens, then it will be this will be called respiratory acidosis. And if accumulation of or if washout of carbon dioxide is there, then it is called respiratory alkalosis. So respiratory disorders are caused, the primary change in the respiratory disorders is the change in the pCO2 levels. And um, the metabolic disorders, on the other hand, are called by abnormal processes which alter the pH by primary change in the bicarbonate concentration in the blood. So if the bicarbonate concentration decreases in the blood, the condition will be called metabolic acidosis. And if the bicarbonate concentration increases in the blood, the condition will be called metabolic alkalosis. Now, respiratory alkalosis occurs because the we saw that per day the human derives 12,000 to 14,000 millimoles of carbon dioxide, and this is excreted out by the lungs. So most cases of respiratory acidosis are due to decreased alveolar ventilation. Now, decreased alveolar ventilation may happen in some neuromuscular abnormality. If the respiratory membrane is thickened and no diffusion is taking place in comatose patients, and many, we are not going into details of that. But the main thing is that in respiratory acidosis, there will be excess amount of carbon dioxide accumulated in the blood. So, what will be the compensation? See, if there is metabolic acidosis, the first compensation will be by respiration. But if the abnormality is there in the respiration itself, there cannot be any compensation other than the kidneys gearing up and making the corrections. Okay, so respiratory acidosis compensation will be by the kidneys. So the kidneys, what will the kidneys do to excrete the hydrogen ions? There are increased in acidosis, there will be increased hydrogen ions. So increased hydrogen ions will be secreted into the lumen. And we know that carbon dioxide is required for hydrogen ion secretion. Carbon dioxide will combine with water to form carbonic acid, which will form hydrogen ion and bicarbonate ion. So if the PCO2 level is increased, then the CO2 level in the PCT cells will also increase. So more of the hydrogen ions will be secreted to reabsorb the bicarbonate ion and to be excreted out. So there will be complete bicarbonate reabsorption, number one, and there will be increased secretion of hydrogen ions in the renal tubules to generate new bicarbonates. There will be increased titrable acidity because more of the acid is being excreted, and increased ammonium in the urine because the kidneys will generate new ammonium because it has to uh, give, or it has to excrete more hydrogen ions in the urine. So the ammonium level will also increase. Metabolic acidosis, it is a condition when the fixed acids in the blood increase. So this can happen either by bicarbonate, if in severe diarrhea when there is loss of bicarbonate ions, or it can happen in other conditions like diabetes is the commonest condition in which diabetic ketoacidosis happens, in lactic acidosis happens, starvation also there is ketoacidosis. So there are a lot of conditions when metabolic acidosis occur. The defect would be fall in the arterial bicarbonate concentration. So the bicarbonate concentration normally is 24 milliequivalents per liter, and if it falls below that, the condition will be metabolic acidosis. So what will be the compensation? Here, because respiratory function is functioning normally, so the first compensation will be from the respiration. What will happen? Respiration can compensate only by increasing or decreasing the ventilation. And what do we want here? Hydrogen ions are more. So we want to expel more carbon dioxide. And we know hydrogen ions increase the ventilation. So the respiration compensation occurs by increasing the ventilation and washing out of carbon dioxide. So what will happen at the levels of the kidney? There will be complete bicarbonate reabsorption. There will be increased secretion of hydrogen ions from the renal tubules with new bicarbonate generation, increased phosphate buffer used, so increased titrable acidity in the urine, as well as increased amount of ammonium in the urine because acid has to be excreted out and we need more buffers. And this increase in ammonium formation for increasing increasing excretion of acid is called adaptation of ammonia by the kidneys. Now coming to respiratory alkalosis. Respiratory alkalosis is a condition in which the arterial PCO2 levels become lower than normal, and it happens in increased ventilation. Increased ventilation can happen at high altitudes, where there is which is a trigger for increased ventilation. It can happen in hysteria fits also. So this respiratory alkalosis is there. And what will the kidneys do in respiratory alkalosis? Low PCO2 will be there, so the hydrogen ion secretion will be depressed. So the bicarbonate reabsorption will be depressed and bicarbonate is excreted, lowering the pH to normal. Because in acidosis, we need to conserve the hydrogen ions, and since PCO2 is low, so the PCO2 is less available to the proximal convoluted tubular cells. Hydrogen ion secretion is low, and when the hydrogen ion secretion is low, the bicarbonate will not be reabsorbed completely and it will be excreted out, compensating for the respiratory alkalosis. What will happen in metabolic alkalosis? Metabolic alkalosis is a condition when the bicarbonates are retained in the body. So the bicarbonate level and the pH will increase. The compensation will be respiratory because respiration is normal. So the respiration will be suppressed because of the low hydrogen ion concentration. But and this can elevate PCO2, but this can happen only up to a limit, because when the PCO2 level increases or when the oxygen level decreases, the chemoreceptors will be stimulated, and this mechanism will fade. So most of the renal hydrogen ion in this condition will be used to reabsorb the filtered bicarbonate. So once the filtered load of bicarbonate, because the bicarbonate level here is more than 24, it may be 30, it may be 40. So the kidneys in the PCT will reabsorb up to 26 to 28 milliequivalent per liter of bicarbonate, and rest will appear in the urine. What will happen in the later segments of the nephron, in the ICT or in the distal tubules and in the collecting ducts? No hydrogen ion will be secreted there, so there will be no titrable acid or no ammonia will be present in the urine. So because no hydrogen ion is available to be excreted out in metabolic alkalosis. This finishes my talk, and I have some questions. I think, can I take it, Dr. Nayak, if you say?
Madam, I think
We
we
should
are
finish.
just end of the stock. Maybe you can just put the questions one by one. If
Okay, okay,
anyone is interested, let them communicate to you.
okay. Sure. So this is the first question, and I would like the students to email me the answers. So this is question number one. Oh, this is already giving the answers. Right? Okay, this is question number two. Answers already there. This is three. Okay, this is four. Thank you so much.
Thank you very much, madam.
Thank you.
We had a very good session today. It's really, really good one, and I believe with the asynchronous presentation, people will be very happy to go through this again and again. And
Thank,
it's really tough. It's really tough, and your presentation was so nice. But it's really tough for the particularly our targeted UG students to have these concepts directly by single chance. So the asynchronous presentation, they can see these things again and again, and they can enjoy the things, and they can understand the concepts. Thank you very much, madam. It's
thank
really,
you so much.
really
Thank you so
honored
much.
to have you here in the stage. And I also thank Dr. Rakhi and yourself for this presentation. Dr. Rakhi has done a marvelous job to give the glimpse of excretory system, which will be helpful for the students to have a before exam, just to see what is there, how things. So that can be done by that part, first half of the presentation. And the second half, you have done a splendid job to explain the concepts. So it will be like, I believe it will be viewed again and again. Those who want to learn the concepts, they want to brush up their concept, they can view this part again and again. Thank you very much. And because we are short of time, we will be finishing this session. Thank you, the speakers. Thank you, our technical team, Dr. Sampa, Dr. Binoy, and the other education committee members who were behind the screen and making this presentation smooth and presentable. And also, I thank our members of Physiological Society of India, executive body and general body members. Thank you one and all. Just before finishing, I would like to announce that our session 5 of the online lecture series will be telecasted, will be broadcasted on November 25th, 7 to 9, the fourth Saturday, November 25th, 7 to 9 evening. And our topics for that session 5 is hematology. Like as we are following, one will be basic, glimpse, overview, and then we will be having in the second half, we will be having applied part. In hematology also, we will be having an overview of the hematologic system, blood chapter, what is required for the UG part, UG basic as well as UG medicine, dentistry, undergraduate courses, whatever required from the blood chapter. And then we will be having applied part at the second half. So thank you everyone. Thank you all the viewers. Thank you madam. Thank you Dr. Rakhi. Thank you Dr. Sompa. So thank you one and all. Enjoy the day and have a happy weekend. Festivities are there, so enjoy. Best wishes for everyone. Thank you. Thank you everyone.